Hello, in this video we are going to talk about the microorganisms. Microbes dominate the planet. Without them, life would cease to exist. They are our past because they are holding the secrets to the origins of life, and they are also our future because they are sustaining life by maintaining essentially all the biogeochemical cycles. The effects of microbes on their environment can be beneficial, harmful, or also inapparent for human observation. Microbes are small organisms. They can be unicellular and microscopic, such as bacteria. They can be colonial and macroscopic, such as protozoans, and they can be also acellular and microscopic, such as viruses. Microorganisms include bacteria, archaea, protozoa, microscopic algae, microscopic fungi, and also viruses. The size range of microbes is immense, going from 1 micrometer to 1 millimeter. The smallest can be a bacteria that measure less than 0.5 micrometer, and the largest are some protozoan cells that are greater than 1 millimeter. In the coming sections we are going to see the prokaryotic microorganisms that are falling in the two domains, bacteria and archaea. We are going to see some of the similarities or differences between them. Let's talk about the shapes of prokaryotic microorganisms. In general, prokaryotes come in various shapes, but many fall into three categories. You can see this figure. We have Gucci, and it is spherical form, bacilli, and it is rod shaped, and also spirally, and it is spiral shaped as it seems in this figure. The diversity of archaea and bacteria has deep connections to their ecology and mode of evolution. For example, when we see the environments and habitats of this prokaryotic microorganism, we can say that archaea are extremophile. This means they can be found at extreme temperature, for example in hot springs. They can be found in extreme salts, for example in the lakes, or also at extreme pH. They are also mesophile. This means we can find them in moderate temperature. For bacteria, they are ubiquitous. We can find them in the air, in the soil, water, in animals, plants, inside human also, etc. The cell wall structure of prokaryotic microorganisms is differing between archaea and bacteria. For example, in bacteria, the cell wall is composed of peptidoglycan, or what we call also murine. In archaea, we find the pseudopeptidoglycan, or what we call pseudomurine. It's called in this way because it looks like the peptidoglycan in bacteria. It's composed of 50% of glycan and 50% of peptide. In archaea we can find also other cell wall structure, for example the S layer, the glutaminoglycan, and the methanoconjunctan, etc. In the cell membrane of the bacteria, the lipids are linked via ester molecule, but in archaea, in the cell membrane, the lipids are linked via ether molecule. When we talk about genome comparison for example, RNA polymerase, DNA replication, transcription and transition, for bacteria, they are unique to this unicellular organism, so they are bacterium. For Archaea, they are similar to eukaryotes and they are so called eukaryotic-like. Let's see other criteria, for example, pathogenicity and sensitivity to antibiotics. For bacteria, it can be. It means that some bacteria are pathogenic and others are sensitive to antibiotics. But Archaea, until today, there are no Archaea that were found to be pathogenic, and there are no Archaea that were found to be sensitive to antibiotics. We are at the end of this presentation. Thank you very much for your attention and see you soon.